Hello, dear students. Today we are learning about regulation mechanisms of plants and animals. Before the end of this lesson, you should be able to identify and describe sensory structures in plants and animals, explain how organisms convert stimuli into responses, and compare regulation control in both groups of living things. We will try to answer this essential question: how do plants and animals detect, process, and respond to stimuli, and why are these abilities essential for survival? Plants are sensitive to a wide array of factors. This includes light, heat or cold stress, gravity, and even the passage of time through day length and seasons. They also detect physical stimuli like wounding by herbivores, infection by pathogens, and mechanical touch from wind or objects. Internally, they use chemical signals to coordinate these responses across their entire structure. One of the primary ways a plant responds to its environment is through a tropism, which is a growth response toward or away from a stimulus. These responses are regulated by plant hormones, natural chemical substances that act in very small amounts to influence cell division, elongation, and ripening. Plants sense gravity. Even if a plant is placed horizontally, the shoot will eventually grow upward, a process known as gravitropism. Phototropism is growth in the direction of light. It is caused by greater cell elongation on the shaded side of the stem, causing the plant to bend toward the light source. Some plants, like sunflowers, exhibit solar tracking by following the sun's movement across the sky every day. Other plants, like beans, have sleep movements, where their leaf blades are oriented horizontally during the day to capture light and shift to a vertical position at night. Light also fundamentally changes a plant's physical structure. Seedlings grown in the light are green with developed leaves, while those grown in the dark have pale, elongated stems and tiny, underdeveloped leaves as they stretch to find a light source. The hypersensitive response is a sophisticated defense mechanism used to stop the spread of pathogens, like fungi or bacteria, throughout the plant. When a pathogen infects a leaf, the plant detects effectors, proteins from the pathogen. In response, cells near the infection site produce antimicrobial molecules, seal off the area by modifying their cell walls, and then intentionally destroy themselves. This creates necrotic lesions, or rings of death, which are regions of dead tissue that starve the pathogen of nutrients and prevent it from spreading. Thigmotropism is a plant's directional growth response to touch or physical contact with an object. This mechanism allows plants to navigate their physical environment and secure structural support. Apical dominance is a regulatory process in plants where the main central stem, the apex, is dominant over other side stems, controlled by the interaction of two key hormones, auxins and cytokinins. If the shoot tip is removed, the source of auxin is eliminated, leading to low auxin levels. This shift allows the lateral buds to become active and develop into new shoots. As seen in the example of a rosemary plant, removing the terminal bud promotes a bushier appearance, because the plant is no longer focusing its energy solely on upward growth, and instead develops multiple side branches. Auxin also plays a critical role in the formation of roots, particularly adventitious roots, roots that grow from non-root tissue like stems. When stem cuttings, such as those from honeysuckle, are placed in a solution with a high concentration of synthetic auxin, they develop many adventitious roots. Ethylene is a gaseous plant hormone that triggers a specific growth maneuver known as the triple response. This is typically seen in germinating seedlings, like pea seedlings, growing in the dark as they encounter obstacles in the soil. The response consists of 3 distinct growth changes. 1. The plant stops growing upward as quickly. 2. The stem becomes sturdier to provide more force. And 3. The plant begins to grow sideways to navigate around an obstruction. Ethylene also acts as a powerful signal for fruit ripening. In a controlled experiment, green tomatoes picked at the same time showed a dramatic difference after 3 days. Those exposed to ethylene turned fully ripe and red, while those in the control group remained green. This chemical response allows plants to coordinate the timing of ripening, often in response to internal or environmental signals. Gibberellins are responsible for dramatic increases in plant size and the development of reproductive structures. In plants like Arabidopsis, which normally grow close to the ground in a rosette form, a surge of gibberellins causes the internodes, the stem sections between leaves, to elongate rapidly, elevating the floral buds for reproduction. Gibberellins also directly influence the size and development of fruit. For instance, spraying seedless grapes with gibberellin during their development results in significantly larger and more robust grape bunches compared to untreated vines. Thigmomorphogenesis is a primary example of phenotypic plasticity, which is a plant's ability to produce different physical traits, phenotypes, depending on the environment it is grown in. This ability is essential for plants to cope with the variability of their surroundings. In a controlled experiment, an Arabidopsis plant that was rubbed twice a day grew significantly shorter and sturdier than an untouched plant, which grew much taller. Because these differences are caused by the environment, the physical stress, rather than different genes, it is a clear manifestation of phenotypic plasticity. Phenotypic plasticity are responses to abiotic factors, neighbors, or herbivory that result in genetically identical individuals having very different physical appearances. The leaves of Passiflora tuberosa exhibit distinct color and pattern variations, depending on the amount of light they receive. The color of hydrangea flowers is not fixed by their genetics, but is instead determined by the chemical composition of the soil in which they are grown. By producing different phenotypes in response to abiotic factors, plants are better equipped to interact with and survive in their specific environments. Now let us look at animals' hormonal mechanisms. Hormones, from the Greek hormon, meaning to excite, are chemical messengers secreted by the tissues and organs of the endocrine system. These molecules signal other cells and are one of the two primary systems for communication and regulation in animals, the other being the nervous system. In crustaceans, such as the Puget Sound king crab, hormones regulate the distribution of pigment within cells, allowing the animal to match its background for camouflage. Male and female elephant seals, Moronga angustirostris, serve as a primary example in the sources of how hormonal concentration and differing cellular responses shape an animal's body and behavior. While both male and female elephant seals possess the hormone testosterone, they differ greatly in several ways. 1. Males are significantly larger than females and are the only ones to possess the prominent proboscis, the large nose, for which the species is named. And 2. Males are much more territorial than females, particularly during the mating season when they use their proboscis to emit loud, intimidating roars. These differences are driven by testosterone, which circulates in the blood of both sexes, but is found at a much higher concentration in males. A single hormone can cause different responses, depending on the tissue it binds to. In male elephant seals, testosterone affects different target cells in specific ways. First, in the juvenile proboscis, it causes the cells to elongate, leading to the enlargement of the nose. Second, in the testes, it signals the cells to produce sperm. Essentially, although the hormone circulates throughout the entire body, it only triggers these specific physiological and behavioral changes in the cells that have the appropriate receptors to bind it. While hormones are secreted into the bloodstream and circulate to every part of the animal body, they do not affect every cell they encounter. A hormone only elicits a physiological response in specific target cells. This specificity is determined by the presence of a matching receptor on or inside the cell that can bind the hormone molecule. If a cell lacks the appropriate receptor, it remains unaffected by the circulating hormone. Remember this: a single hormone can trigger a wide range of activities to coordinate a response to a stimulus. A primary example provided is epinephrine, the hormone behind the fight-or-flight response. In liver cells, epinephrine binds to beta receptors, causing glycogen to break down and release glucose, raising blood sugar levels for energy. In smooth muscle cells of blood vessels supplying skeletal muscles, the same hormone binding to beta receptors causes the cells to relax, dilating the vessels to increase blood flow to the muscles. In smooth muscle cells of blood vessels supplying the intestines, epinephrine binds to alpha receptors, causing the cells to contract, which constricts the vessels and decreases blood flow to the digestive system. Here are specific examples of how hormonal regulation and chemical signaling control animal development and behavior. These examples build on our previous discussion about how hormones coordinate complex transitions, like metamorphosis and social communication. Hormonal processes allow insects to grow and transform from larvae to adults. Asian army ants, Leptogenes distinguenda, use their antennae to detect pheromone-marked trails. In frogs, the hormone thyroxine is specifically responsible for the resorption of the tadpole's tail as it develops into its adult form. The human endocrine system consists of several major glands that release hormones to regulate growth, metabolism, reproduction, and stress responses. Hypothalamus and pituitary gland act as the central control, releasing hormones like growth hormone, oxytocin, and antidiuretic hormone. Thyroid and parathyroid regulate metabolic processes and blood calcium levels. Pancreas produces insulin and glucagon to manage blood glucose levels. Adrenal glands release epinephrine for stress and glucocorticoids. Gonads like testes and ovaries produce androgens, estrogens, and progesterone for reproductive development. Abnormalities occur when these glands produce too much or too little of a hormone. Goiter is an abnormal enlargement of the thyroid gland. It typically occurs when the thyroid is unable to produce enough thyroid hormone, often due to iodine deficiency, causing the pituitary gland to send more signals to the thyroid to grow, or it can be caused by hyperthyroidism where the gland is overactive. Robert Wadlow, the tallest man in history, reached a height of 2.7 meters because his pituitary gland secreted an excessive amount of growth hormone during his development. In the next video, we will talk about the nervous systems, some animal receptors, and thermoregulation in different representative organisms. Thank you for listening.